Welcome to this lecture on cell and molecular biology. Today, we are diving into one of the most fascinating and crucial processes inside your cells, the RNA splicing mechanism. Imagine if every time you wrote a sentence, you included random gibberish that had to be perfectly erased before anyone could read it. That is exactly what your DNA does, and splicing is the cellular eraser. We will explore how this works, why it is essential for complex life, and the mathematical logic behind it. Let us get started. To understand our topic today, imagine you are a famous movie director who just finished filming a massive blockbuster. During filming, you shot hours of raw footage, but not all of it is good. There are bloopers, long pauses, and scenes that just do not fit the story. To make the final movie, you have to sit in the editing room, cut out the useless footage, and stitch together only the best scenes. In the microscopic world of your cells, a very similar editing process happens every single second. When your DNA is first copied into an RNA message, it includes a lot of interrupting sequences that do not code for anything useful. The cell must act like a master film editor. Now that we have our movie director analogy in mind, let us translate this into formal biological terms. When a gene is transcribed, the initial RNA transcript is called precursor messenger RNA, or pre-mRNA for short. Think of this as your raw footage. This pre-RNA contains two distinct types of regions. First, we have exons, which are the actual coding regions that carry the instructions to build a protein. These are your final movie scenes. Second, we have introns, which are the non-coding intervening regions, the bloopers. The RNA splicing mechanism physically removes these introns from the pre-mRNA transcript and covalently joins the exons back together in a continuous sequence, creating the mature messenger RNA. Knowing what needs to be cut is only half the battle. We also need to know what does the cutting. This remarkable feat is performed by a massive molecular machine known as the spliceosome. You can think of the spliceosome as the automated editing software of the cell. It is composed of specialized RNA protein complexes called small nuclear ribonucleoproteins, commonly referred to as SNRPs. The spliceosome is incredibly precise. It scans the pre-mRNA and recognizes specific nucleotide sequences that mark the exact boundaries of the introns. Once it finds these boundaries, it precisely cuts the RNA backbone to release the junk sequence, ensuring that not a single useful nucleotide from the exon is lost in the process. Let us visualize exactly how this molecular machine operates. On your screen is a schematic of the splicing process. Notice the blue blocks labeled exon 1 and exon 2. These are the coding regions we want to keep. The thin line between them is the intron. The spliceosome binds to the 5' and 3' splice sites at the edges of the intron. Instead of just chopping it out linearly, the machinery bends the intron, attaching one end to a specific point in the middle, labeled A. This creates a loop structure called a lariat. The lariat is then cut away and degraded by the cell, while exon 1 and exon 2 are ligated or glued together to form the continuous mature mRNA. Seeing this complex process, you might wonder why cells bother having introns in the first place if they are just going to spend energy cutting them out. The answer lies in evolutionary flexibility and a brilliant concept called alternative splicing. Because genes are broken up into these modular exon pieces, the cell does not have to use every single exon every time. It can choose to stitch them together in different combinations to create entirely different proteins from the exact same gene. If we did not have RNA splicing, our bodies would need a separate gene for every single protein variation. This would make our DNA impossibly large and difficult to manage. Splicing is absolutely crucial for complex life. Building on the importance of alternative splicing, we must also understand the consequences when this system fails. Because the spliceosome must cut the RNA backbone at the exact right nucleotide, the process requires extreme precision. If a genetic mutation alters the boundary sequences of an intron, the spliceosome might cut in the wrong place, accidentally removing an exon or leaving an intron behind. These mutations in the splicing mechanism are not just minor errors. They are responsible for numerous severe human diseases. For example, certain types of muscular dystrophy and various forms of cancer are directly linked to splicing failures. This highlights just how vital this precise editing process is for our daily survival. While RNA splicing is a biological process, we can represent the physical lengths of these molecules using simple mathematical equations. This helps us understand exactly how much genetic material is being removed and retained. The formula on screen shows how to calculate the final length of the edited message. L mature represents the total length of the final mature messenger RNA in nucleotides. L pre stands for the total length of the original precursor mRNA transcript before any editing has occurred. Finally, the sigma term represents the sum of the lengths of all the introns that are cut out by the spliceosome. Physically, if the number of introns or their lengths increase, the amount of material subtracted increases, resulting in a shorter mature mRNA. Now that we have our formula, let us look at a basic numerical example to see how this works in a real cell. Suppose a specific gene is transcribed into a pre-mRNA molecule that is exactly 5,000 nucleotides long. Scientists discover this transcript contains 3 introns, measuring 1,200, 800, and 500 nucleotides. We need to determine the final length of the mature mRNA. Our strategy is to first calculate the total amount of non-coding material that needs to be removed. We add the 3 intron lengths together, giving us 2,500 nucleotides of junk sequence. We then subtract this total from the original 5,000 nucleotide transcript. The final answer is 2,500 nucleotides, meaning exactly half the original transcript was discarded. Moving beyond simple length, let us mathematically model the incredible diversity generated by alternative splicing. To find out how many unique mature mRNA sequences, or isoforms, can be generated from a single gene, we rely on basic combinatorics, specifically the fundamental counting principle. The formula here states that the total number of isoforms, n isoforms, is equal to the product of the choices available for each exon, represented by C. If an exon is mandatory for the protein to function, it offers no choice, so its value is 1. If an exon is optional, meaning the spliceosome can either include it or skip it, it presents a binary choice, so its value is 2. By multiplying these choices together, we find the total functional capacity of the gene. Let us apply this combinatorics principle to a real-world scenario. Imagine a gene that contains exactly 4 exons. The cell's splicing machinery always includes exon 1 at the beginning and exon 4 at the end to ensure the protein functions basically. However, it can choose to either include or skip exon 2 and exon 3 independently. We want to find out how many unique proteins can be generated. Since exon 1 and 4 are mandatory, their probability of inclusion is fixed at a value of 1. Exon 2 and 3 are optional, presenting a binary choice of 2 each. Multiplying these together, 1 times 2 times 2 times 1 gives us a final answer of exactly 4 unique combinations. Practically, this means the cell creates 4 entirely different proteins from just one gene. We have covered a lot of ground today, so let us review the key takeaways. First, RNA splicing is the essential cellular editing process that removes non-coding introns and joins coding exons to form mature mRNA. Second, we learned that this process is driven by the spliceosome, a complex molecular machine that ensures our genetic messages are perfectly readable. Third, by allowing for alternative splicing, this mechanism gives our cells the incredible ability to generate a vast diversity of proteins from a relatively small number of genes. Finally, we saw how simple mathematical principles can quantify both the physical length of the edited RNA and the combinatorial power of alternative splicing. Understanding this process is fundamental to grasping how complex life operates. Great job following along.